Thank you and good morning everyone, and I'm very honored to be here at the first session at the EAACI of the International Eczema Council, which we hope to make a good collaboration in the future as well. I joined as a counselor last year when we were in the American Academy meeting, and I was quite impressed by the level of science and the passion that they have for education and for networking with the other specialities. So I'm really grateful that they are opening to allergy specialty, and I hope that we will recruit more allergists in the future into this very nice network that they have. So my assignment for this morning is to set the scene, because if you are talking about safety of these newly introduced drugs, for us is not so much experience working with them as allergists, so we have to learn from our colleagues which have a little bit more experience. This is why we set the session focused on safety. So I would like to introduce you a little bit to the mechanisms and consequences if we target this particular pathway in the immune system. These are my disclosures. None of them are relevant for the talk that I'm going to give today. So I'm willing to discuss with you this morning where and how we can intervene in the Jak STAT pathway. A little bit about the relation of targeting this pathway and the concept that we call immune fitness. As you know, the immune system is very helpful with fighting infections, fighting cancers, fighting autoimmunity, fighting allergy as well. So we have to think of what the consequences of what we are doing. And then a little bit about the role of this pathway in atopic dermatitis, in asthma, which is new. It's not yet in current practice, but it will come. So I think that as allergists, we also have to understand where and how we can do this in asthma. And then, because this is my personal field of research, how to integrate with personalized endotype-driven decisions targeting this pathway. So, as you know, this pathway is a central communication node in the cell function. Why it is like that? It's because it's stimulated by a lot of substances. Besides, we are very focused on inflammation, and of course we think that and we all know that it's the common language for most of the interleukins. But there are a lot of other signals to this pathway coming from normal, components of our biology, like for example hormones or interferons, colony stimulating factors, and last but not least, biophysical forces influencing the cell. So that means that this pathway can have a lot of good functions, for example, involved in hematopoiesis, involved in the concept of immune fitness, that I will come back to that, in tissue development and repair, and in barrier function, which we know is very important in general starting the allergic diseases, chondroprotection, osteogenic differentiation, and many other more beneficial pathways that are there. On the other side, it promotes inflammation. We know it's a key pathway to promote inflammation, apoptosis, and on the metabolic side, adipogenesis, and it's a very important pathway, for example, driven through biophysical forces into cardiomyocytes hypertrophy. So a lot of research being done on this double-edged, role that this pathway is having in the immune system. Now, if we look at the specific structure of this pathway, there are 4 molecules that are important there, JAK1, 2, and 3, and tyrosine kinase 2. They all have a pretty similar structure with 2 domains. One is the kinase domain, where the JAK inhibitor is acting, which links the ATP and generates the reaction. It's held by the pseudokinase. And then 2 other important components of the molecule, which not only anchor it into the cell membrane, but ensure communication with the signal that comes from outside the cell. Jak inhibitors are expressed in a variety of cells and tissues. I'm not willing to go through this very nice scheme that EMA drafted recently, and I invite you all to look at this paper, which is really, really very informative. Just to let you know that for each of the cytokines, for example, you have a different pathway, and this is where selective inhibition based on the endotype that you would like to target could happen. So this is something that I would like you to keep in mind for our discussion further. Now, of course, being such a powerful node in cellular communication, there are always, mechanisms that counterbalance this activation, and some of them are here on this slide. So for example, starting from the molecular activation at the cell membrane, the CD45 is a very important molecule. Then we all know that for cytokine activation, the SOX is a very powerful inhibitor. Also for the JAK system itself, the PTPs are also important because they do dephosphorylation and they decouple after the initial activation the system. Now, how can we target this pathway? We can think of different levels of targeting, starting, for example, at the cytokine level that promotes inflammation, for example, using recombinant cytokines or blocking the cytokine or blocking the receptor where the cytokine is acting. Then comes the real target on this pathway with the use of JAK inhibitors acting at that kinase portion that I was mentioning. Or we could use, for example, the phosphorylation using peptide inhibitors or small molecule inhibitors, and then at the level of the transcription at the nuclear level, a lot of other interventions like decoy oligonucleotides, antisense oligonucleotides, and small inhibitory RNA. Well, that's the general overview of this. I'm sure there is more, but we are not at the immunology course here. So I would like to talk to you, because now we are slowly starting to talk about and think about the safety of this pathway. What about this pathway and the roles, or the physiological roles of the immune system, which are called the immune fitness? So let's look first at immune surveillance. Immune surveillance, which is, for example, extremely important against infections. And here type 1 interferons are one of the most important molecules. And the good news is that they do have a different pathway of activating this system, providing survival signals, antigen processing and presentation and proliferation, which are good things, together with antiviral response, immune regulation, antigen presentation as well, and checkpoint proteins. This is completely different from the other type 2 interferons, so the pathway is completely different through different signaling molecules which regulates the inflammatory response and the immune regulation. So we do have some possibility of selectivity here in terms of, guarding the good protective anti-infectious immune response and blocking the inflammatory pathways. The second part of the immune surveillance, of course, is looking at the tumors. And here again you have a very, let's say, good dichotomy between this system, with STAT5 being more and STAT3 being more involved into tumor growth and metastasis, and STAT3 being the ones that is helping the immune response against tumors. So again, we're doing MHC presentation and immune system activation, and of course it is harnessed now for speeding up the therapeutic response in anti-tumor immune modulation. So again, we do have a possibility of dissociating here and keeping the protection of the immune response that is through this pathway. Then something that is really recent and is really very nicely shown here, and again we have to have in mind that it's not only the effect on the infections or on tumors, but the immune system has a very important metabolic regulatory role, and we are all learning about immune metabolism, which is very, very important currently. So this is a very nice system showing that upon recognition of infection, this molecule, which is called UPD, is signaling to the cell, and the signaling happens to be through Jak STAT pathway, as we could expect, because there are a lot of metabolic molecules signaling through this pathway, as hormones do. What this system is doing is blocking and is inducing in the smooth muscle cell insulin resistance. That means that the insulin is shifted and the nutrients are shifted away from the muscles and driven to the immune system to fight infection. This is a very, very nice, mechanism that is happening, and we have to check the selectivity of these and what we are doing with our Jak STAT inhibition in the future. Now, I'm approaching the two diseases that we are currently treating as allergists, and the most knowledge that we have, because we already have the molecules in the therapeutic area, is the Jak STAT pathway in atopic dermatitis. And here there are a lot of important cytokines that we currently target for the treatment of atopic dermatitis, and I would like to share with you their signaling pathways and the actions that they do. So for example, IL-4, we know it's a very important T2 cytokine that is a signature cytokine for T2 profile. It signals through the path of JAK1 and then STAT3 and STAT6, and we know that it's very important because it induces IgE isotype switching, regulates T2 target Z genes, attenuates expression of important molecules in the skin barrier like filaggrin, loricrin, involucrin, and claudin. Now, the second cytokine, which is pairing with IL-4, and they do a lot of things together, is IL-13. It signals again through JAK1, but it also uses JAK2 and tyrosine kinase 2, so it has 3 of the 4 molecules of the JAK system, and again STAT6 is there. It helps IL-4 in the IgE isotype switching, regulates special target genes for IL-13, is a major inducer of T2, which is independent of IL-4. This is extremely important, and inhibits polarization to Th17, and again acts on the barrier, decreasing it. The third important cytokine in atopic dermatitis is IL-31, which acts through JAK1, JAK2, and recruits STAT1, 3, and 5, and it's mainly responsible for a plethora of pro-inflammatory cytokines, but we also relate it to each, for example, in its clinical expression. IL-22, the next important cytokine in atopic dermatitis, signals through JAK1, tyrosine kinase 2, recruits STAT1, 3, and 5. It's important in phenotypic shift of the keratinocytes in the skin with the pro-inflammatory phenotype, proliferation, and the generation of what is an important feature in chronic relapsing atopic dermatitis, epidermal acanthosis. Of course, impact on the barrier, but also something that is very important, increased susceptibility to Staph aureus colonization. And another sign that is also important, decrease in the nociceptive threshold. Interferon gamma, so now we shift a little bit the phenotype from T2 to other phenotypes that we encounter into the chronic relapsing forms of atopic derm. Interferon gamma signals again through JAK1, JAK2, recruits STAT1 and 3, and this secreting during the chronic stage of atopic dermatitis, as I was mentioning, is very much responsible for the skin thickening that we are observing there, and of course it's the signature cytokine of the T1 immune response. It induces, of course, inflammation and macrophage activation. IL-12, again, acting together with IL-23, recruits JAK2, tyrosine kinase 2, drives the development of T1 responsive, and IL-23 is very important for a development of Th17. They usually occur in the same pot. IL-19 another important cytokine signaling through JAK1 and tyrosine kinase 2, stimulates the production on the other side of T2 cells, and it's somehow important because it bridges Th17 to T2 into atopic dermatitis and ensures their coexistence. I could not go out of this discussion on cytokines without looking at the epithelial-derived cytokines, which we understand that they are super important in all the aspects of allergic diseases, especially by priming the dendritic cells to promote T2 cells, by promoting T2 inflammation and IL-4 production from basophils, and secretion of periostin, which locally activates sensory neurons to trigger histamine with independent itch. So this is another redundant circle for this serious clinical sign that we are seeing in atopic derm, which is mediated specifically by TSLP. IL-25, again acting through JAK2 and STAT5, reduced filaggrin synthesis and is important to restore the barrier function or to decrease the barrier function, to block it, but it reduces the barrier function. IL-33, which is one of my favorite cytokines, acts through JAK2 and STAT5 and stimulates ILC2s to produce T2 cytokines, especially IL-5 and IL-13, and induces IL-31 production and promotes pruritus. Now, of course, this is not the end of it, because as you know, we do have to think what is really, really important in terms of what should we block when we treat patients with atopic dermatitis in using this pathway. So for example, JAK1 and STAT3 is very important for the skin barrier. So when you think of this phenotype, it's very important to think about this specific part of the JAK STAT signaling. And STAT3 mediates beta endorphin, for example, production by keratinocytes, and is very important in itch. So again, if you think of this, you have to think of the correlation, and I will come back to the end into the clinical phenotype that you are observing and think what you would like to achieve by blocking it. What do we have? Of course, there is another molecule which is extremely interesting, which is the neuronal JAK1, which is again promoting itch, which is independent of the STAT pathway. So if you block JAK1, you can also do this independent of accessing the STAT communication in the signal. What do we have approved in atopic dermatitis? We do have two second-generation selective JAK inhibitors which have a greater selectivity towards JAK1. So think about what JAK1 is doing other than the other JAKs, which is UPA and abrocitinib. And then we have baricitinib, which is the first-generation selective JAK1 and JAK2 inhibitors. This is what we currently are using in our patients. Other potential JAK inhibitors that might come are deucravacitinib, which is an oral tyrosine kinase 2 inhibitor, which is already approved for psoriasis, and probably we will hear about clinical trials on this, and brepocitinib, which is a topical tyrosine kinase 1 and JAK1 inhibitor. Now let's switch to asthma, which is more closer to what I treat. So as you know, we have a current understanding of the complexity of what is happening in terms of mechanisms in asthma, with two major pathways being possible, the T2 pathways and the non-T2. We do not call them non-T2 anymore because we know now that they can be either type 1 with activation of inflammasome or type 3 with activation of IL-17. Always the most severe cases, as you also see in atopic dermatitis, are a combination between T1 and T2 or T2 and T17. And there is not only the immune system is always communicating with the tissue, is its partner all the time, so we call them the shared pathogenic pathways. And here you have the resident cell compartment, the epithelial barrier, the mucin production, the airway smooth muscle, the neurogenic inflammation, which is also very important in asthma, and the epigenetic modifications. So where does this fit in terms of the JAK STAT system? So we know that if you have a T1 polarized response, which is also responsible for a series of cases that we see in asthma patients, you have mainly the gas-like element acting for interferon gamma, and then the phosphorylation of STAT4 is very important, driven by JAK2 and tyrosine kinase 2. Different for the T2 polarized response, where you have the phosphorylation of STAT6, which is driven by JAK1 and JAK3. So in these two types of, in these two endotypes of asthma, different signaling that pathways for the using the JAK STAT pathway. So what is currently being tested for asthma is the tyrosine kinase antagonism for T3 asthma, as I was mentioning, blocking the IL-23 and tyrosine kinase path. And then for T1 asthma and bronchial hyperactivity, because we know this is very much dependent on the interferon gamma, JAK1 and JAK2 blocking, and we do have this interesting phenotype of asthma, which we call IL-6 high, with the activation of the inflammasome, and here the block of IL-6 and acting through JAK1 and STAT3 is also being currently tested. Now that's talking about the let's say start of the asthma cases, but usually, and then you also have the muscles, which are very important. So the c-KIT acts through JAK2 and STAT1, and we do have some data on imatinib decreasing airway hyperactivity, muscle counts, and tryptase release. And the block of STAT5 has been shown to be critical for muscle growth and survival. Okay, so when we go to autoimmune asthma, which is the most most severe spectrum of the disease, where you have the formation of these ectopic B cell clusters and generation of IgG autoantibodies, either anti-nuclear type or against the substances that are secreted from eosinophils. There we usually do have this targeting of B cells with rituximab, but not only, because we know that also the B cell is very important in the signaling of B cell generating autoimmunity. The JAK system is also very important, and it can be blocked with the use of JAK inhibitors. Now, how to integrate with personalized and endotype-driven decisions? Here, for example, these are the potential endotypes for atopic dermatitis. I'm preaching to the choir here because you know better than me, but my thoughts of these, because I have shown you that JAK inhibition is very important, especially for Th22 and Th1 driven inhibitors, that probably these cases of chronic remitting relapsing AD would be the perfect candidates for this type of intervention. Well, for the asthma endotypes and teratypes, where we do have, because we do have a lot of molecules that target the T2 pathway, so that's somehow solved in asthma. But for the very severe cases of asthma, which have a mixed complex endotype, type 1 and type 2, or type 2 and type 17, the Jak STAT inhibitors seem to be the perfect candidate and find a niche there. Also, for example, for non-T2 pathways, where we do not have yet a good molecule which is currently being used. Also, looking at the resident cell compartment, because airway smooth muscle and mucin can also be a very amenable target for this type of inhibition. With that, I would like to invite you to join our EAACI Knowledge Hub together with the International Eczema Council Councillor Amy Paller. We invited her to join us into this safety of JAK inhibitors, offering the dermatology perspective. We also have our colleagues from rheumatology explaining their experience, and also our colleagues from the asthma field. And I would like to invite you to study more on this, because you will hear also about the real-world experience. We also have our own research network, where we currently are gathering data on the safety of these new interventions that we have. Thank you so much, and apologies for being a bit late.